Thanks. It's flattering to be here. That is going to be a tough act to follow for all the right reasons. It's very inspirational. Hopefully my talk won't be seen as sort of dry and academic. What I want to do is talk to you about experiences I've had in the more downstream parts of putting medicines into the patients who could benefit the most. And in particular, talk about the design of emerging trial strategy that I've shared with the FDA and a couple of other regulatory agencies. First, talk about my experience acting on pathogenic variants for idiopathic diseases and how one can go about matching drugs. I'll give an example case that involves a family that's represented here, the Grossmans. Then I'll talk about designing trials and what the motivation for these trials ultimately is and give you some examples. These are in very broad categories, not in glycosylation disorders. And then talk about a distinction that's often overlooked when evaluating people for responsiveness to treatments, having to do with defining personal versus population thresholds. So some time ago, there was a lot of debate on what the virtues and benefits of precision medicine were. And there was a leading periodical, Nature Biotechnology, who asked, is personalized medicine finally arriving? And around that time, a number of textbooks were written describing the principles of personalized medicine. Ironically, about 4 years later, the same journal published an editorial, What Happened to Personalized Medicine? Never seemed to arrive. And my suggestion was that simply we didn't have enough evidence to show that it actually benefited patients more so than traditional approaches. So designing clinical trials to show that a drug works is crucial for moving things forward, as you can imagine. So I'm going to talk about an example where we designed a trial to assess the utility of a drug based on the identification of a genetic variant. Now, many diseases, as has been discussed by the previous speakers, have had mutations identified where drugs that could target the specific defects are what is in question and how one can actually apply those drugs with some effect. Many people have had their genome sequenced and have led to the discovery of mutations that ultimately guided some kind of therapy or intervention. I'm going to talk about, again, the Grossman's story and Lilly's in particular. So how does one identify these variants? Well, typically, you take the patient's genome and you sequence it, and you make the assumption, if the disease is rare and potentially idiopathic, that there's some mutation on the patient's genome that is unique to that patient. So you can identify that by comparing that patient's genome to other genomes and just ruling out any that other people have, because they're not likely going to be candidates. If the disease is rare or idiopathic, no one else on the planet should, in fact, have that mutation. So what researchers do is they sort of invoke a filter where they take all the variants in an individual's genome and throw out those that aren't novel and then ask questions about the remaining novel variants as to whether they damage a certain functional element in the genome, whether it's in a gene of relevance to the pathologies that are observed, et cetera, et cetera. And this is not trivial, as many of you know. In fact, a number of people, including my group, have developed tools over the years to perfect this or sort of advance the science behind this. So we've published papers, again, as have many other groups, showing how one could make claims about whether or not a variant is damaging and likely to be pathogenic. We've also published papers describing whether or not a variant is novel. Now, I didn't put this up here just to brag a little bit, but I wanted to point out that we published these things years ago. And what motivated me was not so much in perfecting these, but asking questions about what one does in the wake of identifying a variant. That's where the problems are, as HUD pointed out. Sequencing is trivial in today's techno-driven scientific environment, but making sense of the variants and acting on them is not. So just to relay one experience, again, go with Lily Grossman's story. Lily had a number of debilitating phenotypes and conditions, been written up in a couple of publications. I was one member of a larger group working on her case. Was very happy to work with Jennifer Friedman and two of my former postdocs, Ali Torkamani and Cinnamon Bloss, on this. Ultimately, we applied the kind of filtering strategy that I described before and ended up on 5 or so candidate mutations that we subjected to functional assays to kind of narrow down and convince us we really did have the pathogenic variant in our pockets. And we're convinced that we have at least one variant that contributes to her condition. So what Ollie did in collaboration with a few other people was just ask the question, that mutation does something, but does it do something that we could counteract with a particular drug? So we queried the literature and found out that there was a mouse study some time ago looking at knockouts of a particular gene that harbored the mutation that Lily had, where there was a successful reversal of some of the phenotype with the drug. That drug was available, so we could repurpose it. We could take the drug that was available and ask whether it could benefit Lily based on the biological story that we had built up. In fact, Lily had some severe nighttime tremors. The drug was provided and it actually reduced the tremors. This was published a little while ago. Unfortunately, as I know from the family, there might be a little bit of resistance, so there's plenty more work that needs to be done to advance Lily's case. Since the time of this study, a group, including Christina Waters, who's back in the upper row up there, has put together an ADCY5 Commons group, not unlike the NGLI.org and other organizations that we heard today, which could really advance the science in dealing with people who have ADCY5 mutations, which is what, in fact, Lily has. So how does one advance the science behind testing out whether a particular drug benefits a particular patient, truly personalize the medicine? Well, before I dive into that, let me just give you a distinction. I don't want to offend anyone's intelligence with this slide. It just puts what I'm talking into a context. So traditional clinical trials basically get a large number of people and give some a drug and some a placebo or comparator. And it could be the case that some people ultimately aren't being given a drug that they're going to respond to. If you think of the color coding here, these people are going to get the appropriate drug. These people aren't. The drug is black and they're colored red here. So you then evaluate the response, and you might see that some people who actually got a comparator drug or a placebo actually responded. This is a huge problem in large-scale phase 3 clinical trials that are based on populations. You might see people that show a partial response that's kind of hard to quantify. At the end of the day, you'll just tally up the number of people that respond to the drug and don't respond to the drug, and then make the claim that the drug might have utility in the population at large. But sadly, what is not being addressed in those trials is, did the drug work for any individual in particular? You don't get that kind of information by default. That requires a different sort of study design, one where we have a patient and we might give them the drug and just keep asking the question, are they responding? And should they be ever taken off the drug for whatever reason, record the non-response if in fact they don't have a response at that point. And keep building up evidence that the drug is actually doing something to benefit them rather than the population as a whole. You could imagine giving them a different drug where they don't show a response. So ultimately, we can then nail down the relationship between this drug and their response. If we had a different patient, they might not respond to the same drug that the previous patient did, but respond to a different drug. So you get more measures on a single individual to make the claim that the drug is doing something of biological relevance to them, not to the population at large. You want to determine these responses objectively. This is a great study designed for repurposing drugs, right? So, to be a little bit more sophisticated, how does one carry out these studies? Well, let's say you have some biomarker. It could be tremor, could be just about anything. You establish just how severe the phenotype is, and then you give them a drug. Then should you not be convinced that the drug is having an effect, even though you might see some improvement, if a downward motion here is consistent with improvement, you could wash them out and put them on a different drug to convince yourself that the drug is having an appropriate, inducing an appropriate response. Do that a number of times and then read off which drug appeared to do the trick. Right? Now, this again is not done in large phase 3 trials because you tend to just use a few measures on lots of people rather than multiple measures on a single individual. If you had a different patient, maybe they'd show a different pattern. And in this way, you can kind of gain insight into how to personalize the medicines that might benefit each patient. I've had the opportunity to kind of discuss these trial designs in many different formats, including, as I mentioned before, with the FDA. Issam Zine, who many of you might know, is actually the head of kind of personalized medicine at FDA. Bob Temple has been at the FDA forever and is a real advocate for novel trial designs. The other advantage to these trials is the focus is not on the drug or intervention. It's on the patient's well-being and outcome. This is a huge difference. So what motivates a lot of large-scale phase 3 trials, and I don't want to be cynical here, is whether the drug is going to have utility in the population and serve the market needs of the people developing the drug. That's different than wanting to make objective claims about whether the drug benefits particular patients. Just a fact. Many N-of-1 trials or single subject focused trials have been pursued in the literature. I'm not going to go through these. We have a review about 4 or 5 years ago describing this. These trials can benefit greatly from all the emerging technologies, your iPhones, your Fitbits, all sorts of ways, finger prick blood samples to get biomarkers. All of these can really help gather enough data in any one patient to make the objective claim that the drug is doing the trick and benefiting them. I've had the opportunity to pursue a couple of these. Just going to give you a couple examples. Here we are looking at antihypertensives. There's a truckload of different antihypertensives that could be meted out to someone with high blood pressure, ACE inhibitors, beta blockers, diuretics, et cetera, et cetera. Typically, a physician is in a state of what's called equipoise, where they don't know which drug to give a patient. So what could be done is you just keep trying them and read off which ones do the trick. So we did this with a couple of patients while I was at Scripps, and as you can see, it looks as though this drug B here, which was diuretic, appeared to lower blood pressure more. Now, what was remarkable in this study is that the patient, right during that time, actually lost about 10 pounds. So even though this was wonderful for the patient because she actually normalized her blood pressure, it was pretty bad for the trial because we couldn't say which drug was going to induce a better effect. So more power to that patient. She became more aware of and more health conscious. Another study that we did that was just published recently looked at an older female with a wide variety of disorders and a genetically mediated sleep phase disorder. So she would, sleep during the day and be up at night. This caused all sorts of complications, as you can imagine, in getting a job and impacting mood and whatnot. And she was on a wide variety of drugs. This problem of polypharmacy, throwing everything in the kitchen sink at a patient based on symptoms, is a real problem, as many of you can imagine. So we designed a study to see what drugs or drug combinations appeared to benefit her most. We used some of the devices that I had mentioned before to quantify the degree to which the drugs helped. We designed the study, different doses and whatnot. Ultimately, what was remarkable and what probably got the paper published is if we looked at the quality of her sleep, the quality of her sleep actually was enhanced when she was taken off the drugs. So it looked as though the combinations of drugs she was on exacerbated her condition at some level. We thought maybe we'd find a sweet spot for dosing and a balance of drugs. In fact, it looked like we should just take her off the drugs completely. So for some of the drugs, now this was very personalized. This might not be the case for another patient, given the condition that she had. Now, with these trials, one could go beyond focusing on a particular individual and draw population-level conclusions. So let's say we had a number of people that we subjected to these N-of-1 trials and determined who was a responder to one or another drug and who wasn't. If we had this kind of data, then of course we could ask what they have in common. Do they share a gene? Do they have some environmental exposure, et cetera, and on that basis decide how to treat future patients. So ultimately, these trials can be thought of as bringing out important response phenotypes that the community might not have access to if all we pursued was this standard kind of large-scale population-based trial. Okay, and I'll dive into something related, but more for monitoring. So if you want to know whether a drug is working, how do you monitor someone? Or if you want to know that somebody is susceptible to disease, what do you do? Well, one thing that the community has to be acutely sensitive to is this difference between personal and population thresholds. And it's a very simple concept, but one that, again, the community has not been leveraging or taking advantage of. So let's say we have some biomarker, and for the sake of argument, let's just assume it's cholesterol level. right? You know, when we have 25 people where we measured their cholesterols and we ranked them. So person 1 here had the highest cholesterol level and person 25 had the lowest. Now, what is typically done in the biomedical community is some kind of population threshold is established that is used to guide physicians for making claims about someone's health on the basis of that biomarker. So this might be a level of 200. If your cholesterol level is over 200, a physician might say, you're in trouble, we need to do something about it, give you Lipitor or whatnot. But let's say we kept measuring the cholesterol levels of these people, such that we had multiple measures over time. What we could see is even though this person number 20 down here is way down here, well below this population threshold, we could establish kind of a personalized average for them. So let's say this is a cholesterol level of 130. No one would be, inclined to believe that's in the danger zone of having elevated cholesterol. So we could establish kind of errors associated with that. Now let's say we have these people come back and their cholesterol level is out of the range that they had established previously. Then despite the fact it's not near the population threshold, this might alert us if in fact it keeps creeping up. Could be a sign of subclinical disease. It could be a sign that something is going on wrong with that particular person, right? So the community uses population thresholds. Just to show how useful the concept of personalized thresholds is, I'll just point to you one particular paper. This is kind of an eye chart. I apologize, it's hard to read, but a paper was published a year or so ago looking at CA-125 levels. These are levels that are tracked to see whether someone might have evidence for an ovarian cancer. It's an accepted and approved biomarker. And what this group did was get longitudinal data where the CA-125 levels were measured at multiple time points on people, some subset of which actually developed ovarian cancer. And they asked, how soon could they have captured the ovarian cancer if they used population thresholds versus personal thresholds? And what they found is, if they had used personal thresholds, they would have captured the cancer earlier. And if the question was how much earlier, it could be almost a year prior to when the actual claim that the person had ovarian cancer was. That's a lot of tumor growth that could have been avoided and maybe dealt with early. So personal thresholds could be useful. Now, I'm involved in a study looking at individuals with an incredibly rare condition that predisposes them to cancer. It's called Li-Fraumeni syndrome, and the individuals are defined by having a T53 germline mutation. This is rare, but for females in particular, they almost have a 100% chance of developing multiple cancers throughout their life. So for people that are born with TP53 mutations, if they don't die a pretty hideous death in childhood of an inoperable tumor, then it's quite likely they're going to develop tumors of all sorts throughout their life, suggesting that they need to be monitored closely, right? So we started a project funded by the Tanner Foundation to develop strategies that would leverage personal thresholds to see if there's a health status change that a physician could act on. And you could imagine doing this in the context of clinical trials, and I'll talk about this in a minute. So this is just one inherited rare syndrome, but one that's pretty pronounced. There was a clinical trial done on the utility of early detection strategies for people with TP53 mutations. And this was probably the most eye-opening clinical trial I've ever read about in my life, and I'll tell you why. There were 36 people that were identified as carriers of the TP53 mutation. They then asked these 36 people whether they'd like to have more aggressive monitoring, blood tests like CA-125, imaging, all sorts of things, or whether that they didn't for whatever reason. 18 chose to undergo the more aggressive monitoring, and 18 for whatever reason chose not to have the aggressive monitoring. Either they were fatalistic about their condition, they didn't want to be inconvenienced, who knows what. Then they evaluated the outcomes of the people who had the more aggressive monitoring versus those who didn't. And here's the survival plots. The people who had the more aggressive monitoring were all basically alive at the end of the trial. The people who, for whatever reason, chose not to have the more aggressive monitoring, most of them died. Now, that's despite the fact that many people who got the more aggressive monitoring did develop cancers. They just captured them early enough to do something about it, or at least enhance their survival. So early detection using kind of personalized thresholds may have utility in a wide variety of settings. That's the point. So in the Tanner project, we've defined a protocol to do this on people with P53 mutations. Laura Getz, a friend of mine, is the head clinician for this study, and it's ongoing. Now, just to be a little bit technical here, what kind of signals would one want to identify? Well, if you've established a historical precedent and your value is outlying, like in the example of cholesterol I gave, then that could be a sign that you should come back in and have a more full workup and see what's going on. You could also just see overall trends. So maybe it's unhealthy to have a decrease in a certain biomarker. So you might act on that, building up data that something is going downhill. You could also have changes, dramatic changes. So in the context of the data we've collected so far in the Tanner project, we've identified instances where patients have had kind of outlying values, shown trends of one sort or another, and had sort of changes. Now we're trying to figure out what the clinical and biological significance of this is, but we're using this as a test bed to vet these ideas of early detection and personalized monitoring. In the context of clinical trials, and this is crucial, because the one examples I gave might not be appropriate for all settings. What one would want to do is make the quickest determination possible as to whether a drug is having an effect, or make the quickest determination possible that the drug is not working. So you want to collect data kind of in real time, and as soon as you think that the drug is not working or causing a problem, you want to get that patient off the drug. You don't want to waste time. So some designs where you have fixed intervals for putting people on drugs, probably not the most appropriate for people that are in debilitating or painful conditions, because you'd literally have to carry them forward throughout the thing unless there was some major medical reason to take them off. So what one would like to do is establish some kind of trend for the patient, give them an intervention, and if things are working out, you want to be able to make that claim as soon as possible. So either you can keep using the drug or start using the drug in other patients that might benefit as well. If in fact there's kind of a change in direction, say for the worse in this place, then you want to know that in the fastest amount of time possible. So one can actually design or see what would have happened had you kept them on, or maybe make a therapeutic change if in fact there's enough evidence suggesting things are going awry, et cetera, et cetera. The point about this is you can actually design such trials to be sequential and evaluated in real time to make the decisions as soon as possible. Many historical clinical trials were not done with this kind of sequential approach in mind. It's usually big fixed samples with intervals of a certain predefined time length. Okay, the last thing I want to talk about, and this just happened the other day, and here I want to give props to my colleagues more so than I. I'm flattered to be a part of this. A lot of people are thinking now about stem cell trials, maybe not just creating stem cells and using CRISPR-Cas9 technologies to factor out mutations, but maybe actually use them as sort of a therapeutic modality, put them back into somebody's body, right? Especially with respect to autologous transplants. Now, there's a lot of concern that this is dangerous, and of course it is, and we have to be incredibly careful. With colleague Jean Loring and a couple of my students and former postdocs, we just published a paper where we wanted to evaluate how likely it was that stem cells that are generated in the dish are going to be chock full of all sorts of problematic mutations that came into being when the stem cells were put together. And what we found is that there was nothing obvious that should just immediately say these should never be used. In fact, and I think this, I wanted to point this out, I think these guys kind of overstated things a little bit. There was nothing obvious. That didn't mean that there weren't subtle things that we have to be aware of. But at least there was nothing obvious suggesting that at a first pass these might be safe. Now, I'm not advocating for the use of these sorts of things now, because I said before, all we did was rule out anything obvious, like a big cancer mutation that all stem cells had in common that you'd never think about introducing into somebody's body. However, we do have to look for the more subtle things before we think about this. So some progress is being made, and I would argue that stem cell therapies are probably the perfect thing for kind of these N-of-1 trials, because they do have to be very personalized and monitored in very strict and rigorous ways. So I will just acknowledge the efforts of my lab in my talk and a couple of other people who've chimed in over the years, and then take a question or two if there's time.
Yeah, we have time for one question.
Quick question in regards to personalized medicine and with pediatric patients. There's a lot of times, for example, speaking from our son's experience, every time that he goes to the hospital because of a sickness, illness, or something, he gets his blood drawn. There's a lot of tests done. One biometric would be the liver enzymes. How would you be able to normalize the data in terms of a personalized medicine to counteract any of the normal progression? We all know that in PMM2 specifically, that liver enzymes as children grow older at different rates for different children normalize. How much of it is going to be normalized based on a therapeutic event or a therapeutic drug, or how much of it is going to be done by the natural progression of the disease? Or
Sure. No, those are great questions. And obviously, the type of study that I described does present challenges. So, for example, you don't want to do a pinprick or a blood draw every 5 minutes just to establish enough data to get a personal threshold. So one has to be sane about how you go about establishing these things. But I would argue this is doable. You know, maybe not from Elizabeth Holmes' perspective because of all the problems she sees, but there are emerging technologies where maybe a little pinprick could get you some blood-based biomarkers that you could use and get at more frequent intervals to establish kind of personal thresholds. I do believe that those technologies are emerging despite the controversy with her company.
One more.
I'm just curious how a lot of those N-of-1 trials got initiated. Was it from the researchers? Was it from the families?
It's a great question. I think it's through communities and engagements like what we're having now. You know, I have not always been in clinical trials design. I wanted to point out that my early career was on genomics, just playing with sequence data. But as I said, I sort of came to the conclusion that, you know, as fun and as interesting as that was, it really wasn't leading to what I wanted my biomedical research to lead to, which is actually having something that impacts someone's life. So I started engaging in discussions with people. I was, again, very happy and flattered to have been invited by Gay and Steve to help out on Lily's case, and had to talk about what in fact we could do that was realistic, and that led to maybe thinking seriously about N-of-1 trials. And the real issue, and I'll be brief here because I know we're pressed for time, but I went to the FDA to talk about that. I was invited. They asked me to go. And the real question that they're grappling with is what constitutes sufficient evidence to compel a physician or the community to use a certain drug. That's what it's all about. And the traditional criteria for making those claims was all this phase 3 population-based stuff. And I thought, well, that might not be the case. As Matt pointed out, if you have a rare disease or an idiopathic disease, you're never going to have the denominator large enough to make these population claims. So there's got to be some other way you can arrive at objective evidence in favor of someone's response. And that's through more repeated measures and other sorts of things. And you can, again, with appropriate statistical methods and study designs, reach objective conclusions about whether someone is responding. I believe that. You just have to then get the communities together to talk about design. Of course, you need a drug. So I said these were great for repurposing. It'd take a while to come up with a new drug and then put it through all the appropriate regulatory things. But at least for repurposing efforts, these trials have they should be seriously considered.